In this class, we will only repeat what we have discussed in the last class. And mostly, I think I will have to do something. I have to repeat RNA editing. That will be the only focus. So it is likely to be a shorter one. So we'll start. I think others may join or not. That's irrelevant. So we in this particular lesson, we learned about what happens after RNA is transcribed. A gene is expressed. The first step is expression. Of expression is transcription. That after transcription, there is a certain low degree of processing done. This is true, especially in the case of eukaryotes. So that is because the genes have introns, and those introns should be removed and exons should be spliced. And not only that, there are some more things about some more changes that can happen to RNA. It's not that every RNA is edited. A few of them, a few examples are there. That is, the editing is about changing the sequence. In splicing also, the sequence is changed, and that is mostly about insertions and deletion like thing. And it's mostly about deletion of the introns. In RNA editing, it is about changing the sequence, and if you're changing the sequence, then that will translate to change in protein sequence. Yesterday also I have discussed this, that one gene can produce one protein, and what we now, through splicing, alternative splicing, we have seen that one gene can produce several different types of proteins, right? They are isoforms. They may have related or unrelated functions, or sometimes they may be non-functional as well. And the other way of how one gene can produce multiple proteins, or at least more than one protein, is by RNA editing. And that is what we will discuss. There are two particular examples. One is site-specific deamination. That is basically modification of the nitrogenous base. And the other one is guide RNA-directed editing. So here is I will just stick to the concept and not go into the biology. And it's about an example of apolipoprotein B. The gene has several exons, and in exon 26 there is a codon which is CAA. CAA codes for glutamine, GLN. And that happens in exon 26. That is present in exon 26. So after transcription and splicing, we get a mature RNA which has the CAA here. That is the 2153rd amino acid or the codon. As such, the protein produced, apolipoprotein, is huge, 4563 amino acids. But in some cases, if this mRNA is transcribed and translated, you will end up with a protein as such a huge protein. And it has a certain types of functions. Apolipoprotein is about transporting lipids from different parts of the body to liver. So we can have the transport of the function of apolipoprotein B when it is produced in fully. It is transporting to the liver, whereas the cytidine CAA could be, or the cytosine that is present here could be changed or modified by an enzyme, cytidine deaminase. The deaminase will remove the amino group and on the 6th position, and it will result in the formation of UAA. UAA is a stop codon. It doesn't code for any amino acid. So if ribosome were translating this mRNA and it comes here, this is a stop codon, so that will stop translation. And it produces a truncated protein. It's about 2,153 amino acids, and it has a different function compared to the full-length protein. Transport to intestine. Okay, so one gene is producing two different types of proteins. One is a full-length huge protein, and the second one is just half the size of that. And they have different functions. So the previous one, the enzyme that we have discussed, is about cytidine deaminase. It removes this amino group and it will result in a keto group, and that is nothing but uracil. There are also other ones which is called as ADAR, adenosine deaminase, acting on RNA. So the same amino group here on the adenine could be removed, and a keto group will form, that is inosine. Inosine is the precursor. If you have studied in biochemistry metabolism of nitrogen bases, you will find that adenosine is kind of intermediate from which guanine and adenine can be made. So if you have inosine in the mRNA, then the ribosome could read it differently and produce a different, or add a different amino acid at that location. Okay, so that is another enzyme, adenosine deaminase, acting on RNA. The point is that cytidine deaminase and ADR are acting or changing the base that is already present in the RNA. The guide RNA based ones are a little bit complex and also at the same time amazing. That is, you have a primary transcript, and during that they can be edited by addition of bases into it. And then the original sequence is the one that is written in black, and now you have 4 uridines added into the mRNA, and they would code for, now the reading frame has changed. Previously, you can consider the reading frame to be something like this, GAA, and these two are 2 different codons. But because you have added the 2 uridines are added in this location, now the reading frame has changed from here to here. One second, please. Sorry for the interruption. Yes. So the reading frame changes, and then you will have the formation of a different protein. So let's see the mechanism of how it happens. And here is GAA. We are talking about how 2 uridines can be added in this location, right? The mechanism is similar for the rest of the things. To start with, we discussed that there are 3 different regions for the guide RNA. That is, it has anchor region, it has an edit site determinant, and a poly U stretch. Sorry. Yes. So here is the guide RNA. Guide RNA is providing a template, or kind of deciding where this editing should happen or insertion should happen. There are 2 enzymes that are important for inserting the uridines. But then where it should happen is guided by an RNA, and that is why you call this RNA as guide RNA. So guide RNA has these 3 components: anchor, that has the region of homology, with this, say for example like that, and it has editing region where the editing should actually happen. So these two also uridines are already added, and now you have the guide RNA that base pair something like this, and there are two adenines here which do not base pair, and they are in kind of hairpin loop like this. Okay, so there is an endonuclease. Endonuclease cuts exactly in that location where the hairpin is formed. And now the enzyme which says terminal uridylyl transferase, TUTase, terminal uridylyl transferase, which finds this is the terminal, and uridylyl transferase, which will add two uridines there. That's the name also, terminal 3' terminal uridylyl transferase. And two uridines are added to in that location, and then the ligase enzyme seals the phosphate backbone, forming a complete RNA. So there are three components that are involved. One is endonuclease, that is where it makes a cut at the mismatch where the hairpin is formed. Then you have the TUTase, and then we have the RNA ligase. And these are the ones that determine what should happen at these are the enzymes that catalyze the reactions, and guide RNA, okay, small, is what determines where it should happen. Okay, and then the last is about mRNA transport. We know for all the mRNA, so far what we have discussed in this lesson is processing. We have discussed during transcription itself, we learned about cap, the 5' cap, G cap, methyl G cap. And we learned about splicing of introns in this particular chapter. And also in the previous chapter, we learned about polyadenylation. It happens only in eukaryotes. All of these things we are talking about eukaryotic mRNAs. So the mRNA that is already matured and formed inside should now be transported, because translation happens only in the cytoplasm. Whether it is bound to the endoplasmic reticulum or not, freely in the cytoplasm, it can only happen in the cytoplasm, because that is where amino acids are there, that is where tRNAs can be charged, that is where you will have the ribosomes. Ribosomes are formed in the nucleolus, and those ribosomes will be transported out, and then translation can happen continuously here. So how the question is, there are pre-mRNA and there is mature mRNA. That is, all the processing is done. Only the mature mRNA should come out. It's not between or while splicing is happening. And then how is the selection of mature mRNAs made? What we know so far is exonic splicing enhancers. I hope you remember that. Those are certain regions that are present in the exons to which SR proteins bind. If there are two RNAs, say for example RNA A and RNA B, if this RNA is completely coated with SR proteins, what does that mean? If it is completely coated with SR proteins, that means there are no introns, because SR proteins only bind to exonic splicing enhancers, and exonic splicing enhancers are typically present only in exons. Introns do not have it, right? That means it says that this protein does not have introns if it is completely bound by SR proteins, okay? And also we have the G cap should be there, and also the poly A tail. And there are many other proteins called as exon-exon junction binding proteins, something like this. So these are SR proteins, and you have, assume this is one exon, and you have the next exon binding. The exon will be coated with SR proteins. In the middle of those, the junctions where exon, say exon 2 and exon 3 are bound, there are proteins that bind there, which is called as exon-exon junction binding proteins. So if a mRNA is filled with SR proteins and it has the exon-exon junction binding proteins, then it is completely devoid of introns. And there is one more important protein that is called RAN-GTPase. RAN-GTPase is the one that binds to the 5' cap. So if there is no 5' cap, RAN-GTPase does not bind there. So RAN-GTPase is particularly important for transport of mRNA into the cytoplasm. There are many other factors about how mRNA is transported out of the nucleus, but still we will just consider only this much for the sake of our understanding here. The SR-rich proteins do not have any function in the cytoplasm, and exon-exon junction binding proteins also do not have any function. And RAN-GTPase can also be taken back. These proteins, once they reach the cytoplasm, they dissociate from the mRNA, and then they are taken back into the nucleus, where they will bind to newly spliced mRNAs or mature mRNAs, and then they will bring back. So their function is mostly to transport mRNA from nucleus to the cytoplasm. These are some of the questions that we typically have. I explained the mechanisms of RNA splicing. Please remember that for any of the questions in the final exam, there will not be any direct questions. You will have to think and write. There will not be much to write, actually. You should also practice well for drawing the diagrams, with labeled diagrams and so on. But otherwise, in this lesson, you should be able to recall the different types of mechanisms of splicing. You should know the chemistry, that is the trans-esterification. Then we have studied about 3 different splicing mechanisms. One is the spliceosome or nuclear intron spliceosome mediated ones. Then we have learned about group 2. These two are almost similar. And then we have learned group 1. Spliceosome, there are complex about 150 or so ribonucleic proteins involved, and this happens with the nucleus. Group 2 self-splicing, they do not need accessory components. They can splice. The reaction is almost same as that of spliceosome mediated splicing, but they do not have extra components. And then there is a group 1. And group 1 and group 2, the major differences are in group 1, in spliceosome mediated and group 2, you have something like the branch point A site, and that forms 2' to 5' linkage. In group 1, there is the guanine GTP that is present non-covalently bound to the intronic region, which will eventually form the G will become covalently linked to the 5' side of the intron during splicing. And there is one more. The difference is in these two, there is a lariat formation, whereas in group 1, you will have a linear intronic intermediate that will be degraded eventually. Right? That is quickly, you can think of them as that. There are many proteins. I think you have to remember about the spliceosome mediated one, because that is the complex one. If you remember that, these two are easy and simple. We also learned about alternative splicing. It's about how one gene can give rise to several by including or excluding some exons. There is also mutually exclusive splicing is also there, and so on. There is a diagram with 5. I think you have already done the assignments. Some of you have done the assignments, and I think you have got a good understanding of what it is. And the RNA editing, we have discussed about modification of the bases in the RNA, and insertion or deletions can also be done by using the guide RNA. Modifying, we learned about ADAR, adenosine deaminase, acting on RNA, and cytidine deaminase, and guanidine, guide RNA, through which insertion can happen. It would require endonuclease, it would require TATase, terminal uridylyl transferase, that is 3' terminal uridyl transferase, and it will require RNA ligase. Don't just write ligase. When we learned about replication, we learned about DNA ligase. This one is an RNA ligase. And in mRNA transportation, we just finished that. It's mostly about what covers whether the GCAP is present or not in the RNA, whether SR-rich proteins are coating it or not, and exon-exon junction binding proteins are present or not, and also the poly-A tail. And if a protein like that is there, then the RAN-GTPase can bind here, and it will transport, along with many other accessory proteins, it will transport the mRNA from nucleus to the cytoplasm. Okay, that ends today's class, and I will stop the recording now.